The overall goal of this procedure is to generate stably transduced organoids from primary mouse intestinal epithelium for downstream analysis of intestinal organoids. This is accomplished by first transfecting HEC293 T cells with lentiviral packaging vectors and a lentiviral plasmid encoding the gene of interest to produce high-titer lentiviral particles. The second step is to pre-treat cultured mouse intestinal organoids with a number of growth factors to obtain cystic hyperproliferative crypts. Next, transduce the organoids with the high-titer lentivirus and incubate the transduced organoids in matrigel. The final step is to embed full-grown transduced organoids in paraffin for downstream immunohistochemistry analysis. Ultimately, lentiviral organoid transduction is used to generate genetic alterations in an in vitro model of the intestinal epithelium that is reliable, reproducible, and fast.
The main advantage of this technique over existing methods, such as cancer cell lines, is that organoids are non-mutated, display stem cell hierarchy, intact cellular polarization, and can differentiate into all different cell types of the small intestinal epithelium. In addition, transduced organoids can be used to study specific genetic elements, thereby outweighing the use of transgenic mice in facets of cost and speed.
The production of lentiviral particles is a 5-day protocol that begins with splitting HEC293 T cells to 60 to 80% confluency in a 162 square centimeter flask in cell culture medium. Incubate the cells overnight in a humidified